Hey guys, welcome to Chapter 8, Topic 4. In Topic 3, we just talked about DNA recombination. Now we're going to talk about mutations. These are things that can occur within the genome of the cell. And so it's a little bit different way of looking at how genomes can be changed in microbes, or really in any organism for that matter. So this topic is important because mutations create changes to the microbes, and this can alter the way they function, either if we're looking at them in a laboratory assay or if somebody has an infection of something that is different than normal. So it's really important that we understand how this can happen. Here are our objectives for this topic. Make sure you can understand these, especially number 2. This is going to get a little confusing when we get to that slide, but there's a lot of different types of mutations and they all overlap and they kind of have a different feel to each of them, so it's really important to have a good sense of what these different mutations are. So first of all, what is a mutation? A mutation is any change in the genetic code. This can be a silent mutation, meaning that there's something that has changed in the genetic code, but we don't know about it. And this is really common in humans, for instance. We all have a lot of different changes to our genetic code that may or may not actually have any kind of outward impact. So that's a silent mutation. Then we have noticeable mutations. These ones can create very big disabilities. They can change the color of a microbe that it grows on a plate. It can even be lethal. So there's quite an extreme range on noticeable mutations. One important thing to remember is that mutations are a driving force for evolution. This is how species evolve, is by having a mutation that gives them an evolutionary advantage. And we're not going to go too much into evolution here because this isn't the right place for this, but it is important to remember that mutations are not always harmful. They can be helpful to an organism. And before we move on, we need to discuss two important terms. The first of these is wild type. A wild type organism is an organism that does not have the mutation. This is the one you just commonly find in nature. Basically, it's kind of our control. Versus a mutant strain. This is one that has an observed mutation within it. And one example of this, and you may not even be aware, is that the microbes that you guys are using in lab are mutated organisms. We have bought specifically ones that are a little bit safer for you guys to work with. They're not as virulent. You're not as likely to be able to ingest them and get a disease from them. That doesn't mean they shouldn't be handled with care, but I'm just saying they're not as pathogenic as the wild type versions of them that you would find in a clinical laboratory or out in nature. So what are the causes of mutations? There are two main causes. The first is spontaneous mutation. This is when there's a random error in the DNA replication process. And something that maybe you'd think of with this is like cancer, for instance. If there's a mistake when cells are being copied, the new cells won't have the right genetic information and could develop into a tumor. And so that's kind of where that comes from. Now, generally our housekeeping genes are really good and catch these mistakes, but they don't always catch them. So that's what a spontaneous mutation is. We also have induced mutations, and this is an example of being exposed to a chemical or radiation or something like that that can induce changes to the genome. This can happen with bacteria. For instance, we talked about how UV light can be used to kill off bacteria. Well, if you don't expose them to enough UV light to actually kill the bacteria, it'll actually just mutate their genome and create mutant organisms. So that's where this kind of comes into play. So there's two different types: spontaneous, which happens naturally within the cell, and it's a mistake due to natural cell machinery. And then there's an induced, which is chemical or radiation or environmental exposure based. All right, so this slide's got a lot of stuff on it and it's going to get a little confusing. So feel free to pause and rewind and come back to this explanation a couple times if you need to. First, we're going to start with two main categories of mutations: lethal mutations and neutral mutations. Lethal mutations cause death in the cell. This mutation is so bad for the cell that the cell will die. This can either be, the ribosomes aren't going to fold right or something critical. Maybe the membrane can't be built. It's huge. These are lethal mutations. Then at the complete opposite end of the spectrum are neutral mutations. And neutral mutations produce neither an adverse or helpful effect. So it doesn't hurt them, it doesn't help them. And in fact, it's very difficult to tell if they even have it unless we sequence the genome. So those are two big extreme types of mutations. Now, as I mentioned before, there's middle ground ones, and we're not going to talk too much about that right now. Those are two big extremes for results. Then within these types, we have two types of mutations that can occur. Point mutations, where a single mutation occurs on a single nucleotide base. This is one base. Now, as you've learned from protein transcription and translation, you know that depending on where that mutation is, it could have no effect or it could have a big effect. Could change the amino acid that's coded for it, or it may not. And this comes back to that lethal and neutral mutation. Frame shift will actually cause a huge issue. Frame shift says that instead of reading this set of codons A, B, C, it's going to read B, C, D, and it's going to keep going down the whole way and completely misreading the protein sequence. That's a point mutation. Point mutations have a lot more dramatic effect than a neutral mutation, than a point mutation. So frame shift misreads a whole sequence of things. Point mutation, single nucleotide, may or may not have an impact. So let's talk about different types of mutations within these. So we have lethal and neutral, which then can be a point or a frame shift mutation. And then we have these types of mutations that occur within these types of groups, and they can be in one or both. A missense mutation causes a different amino acid sequence that produces a different shape. Remember, amino acids have very specific structures, and if you replace one of them with something completely different, the protein is going to completely misfold. You have nonsense mutations, and this is where a stop codon gets inserted in the middle of the protein, so the sequence isn't nearly as long. So say the protein is supposed to be 100 amino acids long, it may only be 10 or something like that. So that's a nonsense mutation. Then we have the silence mutation, where the wobble base can be affected, but it doesn't actually change the amino acid. So it's supposed to be arginine, still arginine. Then we have a back mutation, and this is actually where a mutation goes back to what the original sequence is supposed to be. So I know that that's a lot of different terms and gets a little confusing. What I would suggest is consider like a Venn diagram or a chart to help you really sort out these terms, to make sure you have a good handle on these different types of mutations, because since they can all be in one or more categories, it gets a little confusing. So how do mutations get fixed? Mutations can be fixed. We don't talk about them getting fixed because we only usually talk about when they've actually happened and we can see the product of them. But in fact, a lot of mutations get fixed every day in cells. And there's two main mechanisms of doing this. There's a DNA proofreading mechanism that will come along and proofread the genome after polymerase has made a copy of it to make sure there aren't any mistakes. There's also something known as light repair, which can detect when there's damage due to radiation or something like that. What happens regardless is that these sections of DNA are found, and then a process called excision repair through an enzyme complex will come in, remove the wrong bases, and put in the correct bases, and then it will continue on. So this is how they get repaired. So this is why not all mutations are seen immediately, because they get fixed. They get fixed before they become a problem. So to wrap up this topic, as I mentioned earlier, not all mutations are bad. Some of them are positive. The positive mutations are what have resulted in you and me being here today, or resulted in the E. coli that we know, because these mutations help organisms survive in their environment. Now, negative mutations are pretty obvious, right? They make it so that the organism can't survive in that environment, and so they don't survive. So an example of this for a positive mutation would be if a drug had the methicillin resistance gene. Let's say that it was a staph gene with this. If methicillin is not present in the environment, this mutation doesn't do this organism any good. And so it has just as equal likelihood of surviving as not. Now, if the environment starts using methicillin, then obviously these mutated cells are going to have a better chance at survival and further division and carrying on of the genetic information. So that's where these mutations play a role. It only matters if the environment is selecting for these traits. So let me know if you have any questions on that, because it's a little confusing. It's a little bit of an evolution aspect of it, and if we need to, we can spend some more time talking about how those environmental impacts select for or against mutations. So this is the end of this topic. Please review the objectives, and as always, let me know if you have any questions about any of this content.